Somebody joined the link from the first session. 1 second.
So sorry, guys.
All right, we're just missing a few more, but I think I'm going to give 2 more minutes and then we'll jump into things. No issues at all, Nico. No problem at all. I know that connection issues can be challenging. I actually, when I moved back to the U.S., I was living in a pretty rural area of U.S. and I was amazed at how poor the reception the internet was in rural U.S. I remember being in rural areas of southern China where I felt like the cell service was way better than what we get in the rural U.S. So no concerns at all. All right. So I think we have 1 or 2 that'll join in a little bit later, but that's okay. Well, I hope everybody is doing well. I obviously, since you're here, you found the email with the new session link, which is great. And hopefully you saw a few of the resources that I tagged there in the email as well. So I am going to jump straight in because we're going to cover quite a bit of ground today, and I want to be respectful of everybody's time. I just want to remind you again, if you have any questions, feel free to throw them over into the chat. I'll be monitoring that, and then I'm going to. Yep, there we go. Okay, good. Jump over to my presentation here. Okay, cool. Now we're ready to go. So what I'd like to do just very briefly is yesterday, the key of what we focused on was our discussion about what is fermentation. And we discussed through different aspects of defining what fermentation is. And I want to share this again with you because it's going to frame a lot of what we discuss today. We provided some clarity into a strict definition, a strict sense of fermentation is the catabolic, and remember that's the breaking down. So catabolic being when things are broken down and energy is released, metabolic pathway of anaerobic cellular respiration that begins with glycolysis. And today we're going to dive into here in a minute about what exactly is glycolysis, what is happening, and how fermentation impacts and influences that. And then we mentioned the important aspect of both these processes happening externally outside of the coffee by certain actors, as well as internally as metabolic pathways inside of the coffee seed. And we finished our discussion by pointing to the significance in the role of microorganisms in this process, particularly our bacteria, yeast, and fungi. So today, we are going to turn our focus to microorganisms. Today, I'm planning on covering two different presentations. So we'll cover our topic on microorganisms, and then we're going to talk specifically about some aspects of the actual fermentation process. And that's when we'll dive into glycolysis and some other topics. So today we're going to talk about in this lesson we're going to talk about microorganisms. We're going to describe some of the differences between bacteria, yeast, and molds, and we're going to talk very briefly about some of the common microbial species that we find in coffee post-harvest processing. Now, in a later course, a later lesson, we're going to dive into what are the sensorial or what are some of the byproduct characteristics of those different microorganisms, but we're just going to look at them from a just an identification level here in this lesson. So, is there a question? Sorry, I just wanted to make sure if anybody I don't want to be moving too fast. Like I said, I started early this morning, so I loaded up on caffeine. So if I start going too fast, I usually, when I'm in in-person class, I tell people to throw something at me, which that wouldn't work in this context because you would just throw it at your computer. But, feel free to stop me. When we talk about what is a microorganism, we're going to look at a definition of what a microorganism is, and we get lots of these different terms: microbes, microflora, microbiota. We're going to talk about what those are, and then we're going to look at some differences between a prokaryote and a eukaryote. And we mentioned this yesterday when we were talking about some of the taxonomical structures, and it seemed like a little bit of a rabbit trail when we kind of dove into that, but I present that to provide a little bit of context for what we're going to talk about in this discussion about the different types of microorganisms. Antony van Leeuwenhoek, and I totally butchered that name, I know, and my European friends can all laugh at me, and Robert Hooke are the ones who are documented with the first identification of microorganisms. And essentially the name came from the understanding that you needed a microscope in order to view them. And it's funny because this didn't happen until 19th century, and it's interesting to think about how that must have opened up like a whole new world in terms of being able to actually visually see these different microorganisms. And when you go back and you look historically at the progression from thinking that they were animals or plants, and then kind of being able to understand them more and more, it's quite fascinating as kind of a historical progression. And it is also interesting to note that not all microorganisms are only visible with a microscope. So many fungi are identifiable with the naked eye. And there's even a few bacteria. Specifically, there's a few bacteria that will change colors, and so they become, visible to the naked eye. But and in the same respect, we oftentimes refer to microorganisms as, like saying, the microbial communities or microorganisms. And it'd be similar to studying human beings and calling it a study of animal communities, right? We think about, so many diversity of animals and different types of animals and species and et cetera. And in a very, very small microcosm of that, microorganisms represent a huge amount of species and organisms throughout the world. When for this course, in this class, and specifically with fermentation, we're talking about microorganisms. We're focusing specifically on bacteria and fungi. And fungi will break down into yeast and molds. But it's important to note that they both fall under the category of fungi. And again, I'm a fun guy. You might be a fungi, but I'm going to stick with fungi if you'll allow me. So some of the characteristics of microorganisms, generally find that they are unicellular, though there are some exceptions to that. And it's interesting, even in a lot of the preparation and stuff for this course, I've spent quite a bit of time watching different YouTube videos, and there's an academy, and I'll put it here in the chat here. It's called Khan Academy. If you've ever come across this, really, really good resource for scientific just information. And they have some really cool videos and different discussions and so forth. There's also you'll see cited a couple times this a source called the Amoeba Sisters, which is kind of it's like a comical, interesting. I'm sure my children one day will enjoy watching them as they kind of study different scientific information because there's some really cool videos as well. So it's called the Amoeba Sisters. If you're interested to dive into these concepts a little bit more, um, so microorganisms and where I was going with that was it's we're learning more and more, just as you know it was around the 19th century when this kind of world of microorganisms was opened up. As science advances and we continue to study more and more, we are open and exposed to the complexities of what all of this information is and what the world of microorganisms are. So and we understand that these microorganisms can also be alone or part of a colony. Some can be useful, some can be harmful. The complexities are immense, and in fact, the interesting thing here, and this is a diagram from a study that was looking at basically classification of microorganisms based off their function, and it just kind of shows you it's not necessarily important what all of these functional diversities are, but we see vast amount of purposes and uses for microorganisms, decomposing, um, composting organic matter, pathogens, different food and beverages, which coffee will partake in that, the joy of that, waste, mineralization, all of these different types of functions all are represented in different types of microorganisms and their metabolic pathways. Like I said earlier, the main groups, bacteria, fungi, which are your yeast and molds, archaea, and protozoa. Protozoa are an interesting one. We're not going to dive into, as I said, we're going to kind of keep our focus to more of our bacteria and fungi. But protozoa are a group of single-celled eukaryotes, of which a good majority of them are parasitic or parasites. They rely on a host. So just one thing to note about microorganisms. There was a really interesting video that I was watching that was showing kind of a 3D rendering of different types of microorganisms and kind of showing its scale, you know, as it kind of got larger. And, you know, it was part of my nerdy dive into the internet of YouTube videos and interesting stuff on microorganisms. But just how micro are these organisms? You can see here on the right side of the image is an actual, you know, a microscopic view of these different organisms, and on the left is kind of a rendering of them. But you can see that molds can be observed at relatively low magnification. Again, you do have some molds that are visible by the naked eye, but you can see how they are structured and kind of their shape. Yeast there about 100 times, and that's a typo. Bacteria should be even more. So actually what we have here is that molds are at a size classification are the largest. Yeast are then the next smallest, and then bacteria are the smallest. Typically, we measure them in microns, which you can see the symbol or the kind of abbreviation for microns, and you'll see that appear different times throughout our presentation here on microorganisms. So all of these terms, you've heard microorganism, microbe, maybe you've heard microbiota, microflora. To me, it's kind of like the variety discussion. You know, you hear varietal, variety, cultivar, hybrid, heirloom, landrace, what's right, what's wrong. You know, some people have a very passionate opinion about it. Some people, you know, are very free flowing with their terminology. Just to provide a little bit of context for you. Microorganisms, again, initially meant basically organisms that were not visible to the naked eye. And so you can imagine the complexity or the amount, the massive amount of organisms that were defined or constrained within that terminology. But you also see the term microbe. Now, I will say that microbe tends to have more of a negative connotation. And not that it's necessarily true. It's just more kind of traditionally or typically what we see. And so you will find that term used, but again, it's oftentimes like when we're talking about a virus or something that's potentially harmful, oftentimes we use the term microbe. And then you'll find this term microbiota. And this one, I think, is a really important one as we talk about coffee or really fermentation for that matter, because it talks about the microorganisms of a particular environment. And I don't want to dive too deep down this rabbit hole of kind of terroir and kind of those discussions, because that's maybe a conversation for another day. But there is a uniqueness, and research is showing there is a uniqueness of the microbiota that exists in certain parts of the world, full stop. There's different types of organisms, there's different types of species of yeast, of molds, of bacteria, etc., that make up that region's or that area's microbiota. Now, how that might impact coffee flavors or coffee fermentations is obviously going to be dependent on so many factors, but there is some interesting research that looks at the microorganisms as a type of signature or fingerprint for different regions of the world, and that's significantly linked to those microbiota. So you can kind of know that when that terminology is used, it's referring to the specific microorganisms in that environment. You don't see this as much anymore, but you did previously see the term microflora, and it kind of goes back to a very kind of I guess previously held or previously understood view that that all living things were classified into either animals or plants, and microorganisms were in the plant kingdom, therefore flora. That term was used. Now we know that that's no longer true. We've identified it as a separate kingdom, and we talked about that yesterday. But that's where that terminology microflora kind of originates and comes from. Not really the best term to use now, but every so often, especially if you're reading or looking into kind of older research, you'll see that terminology being used. All right. I want to discuss briefly, we've mentioned previously this kind of breakdown of prokaryotes and eukaryotes, and we're going to talk more about that, and we'll reference it, but I just want to give us a little bit of a definition, or at least an understanding, comparison between the two. So eukaryotes are typically multicellular, whereas prokaryotes are going to be unicellular. Again, hold that a little bit loosely because you will find some exceptions in that, but generally speaking, that's true. But one of the key functions and some of the key differences are that a eukaryote is going to have a well-defined nucleus surrounded by a nuclear membrane. I'm going to show you this picture here, and I'm going to jump back to this chart. So if you didn't get everything, that's fine. You can see here an example here of a eukaryote and a prokaryote, and you can see in the middle the blue section here in the eukaryote, and the kind of light blue, sky blue, if you will, kind of image here on the portion here on the right. The eukaryote is going to have that well-defined nucleus surrounded by this nuclear membrane. And then whereas your prokaryote is going to have a very poor defined nucleus and is not going to have that nuclear membrane. So therefore, some of the differences, the way that compounds move in and out of that nucleus, the security of those structures are all going to be impacted by that. In addition to that, the eukaryotes have specific cell organelles like mitochondria, whereas the prokaryote doesn't. And we can see that here in this illustration. You can see the complexity versus the simplicity of the different compounds or of the different organisms, and why you can see how the differences that we see in our domains being prokaryotes and eukaryotes and archaea. Examples of eukaryotes: your fungi, plants, and animals, whereas your prokaryotes being your more bacteria, algae, archaea, and etc. So again, the main point of this is to begin to understand and when the challenge is always when we begin talking about a lot of these concepts, and if we mention terminology and things that we have no experience with or have no reference for, it can be a little bit overwhelming. So a lot of this information is all about just building on that foundation that we kind of progressively kind of work through. All right, so let's turn our attention to bacteria, yeast, and molds. Okay, so we're going to talk about each of these individually. We're going to, for each of these, we're going to talk about some different classifications, some different types, and then we'll talk briefly about some that we find in coffee post-harvest processing specifically. We'll spend a little bit of time focused here. And again, I'll come back to some of this as well in a later presentation when we start to connect some of the different types of compounds that are produced. I think it's also important to just define as well here for a second this idea of metabolites. You hear that word used, and essentially metabolites are those the different types of byproducts that are produced. So a metabolite is something that is produced in that process. And so when we think about, and we mentioned, those byproducts that are influencing coffee, that then becomes a metabolite. And it's just kind of a term used to describe all of those. Tristan, I might be getting ahead of myself. Is a prokaryote more easily affected by its environment because it has a smaller membrane? Absolutely. And you could find that some of those are, and here later in the presentation, I talk about some of those conditions being oxygen, being acidity, being temperature, those other types of changes in our environment that can impact that. You still have some of that influence with eukaryotes as well, but the impact or the potential for impact is greater. So yeah. And good on you for being in the middle of the morning there and being fresh on it. So way to go, Tristan. We're all way impressed. So some of the characteristics of bacteria specifically, we see that these are the largest group of microorganisms. Okay, we typically find that these are prokaryotes. They are among some of the oldest forms of life. Ubiquitous basically just means that they're everywhere. And I mentioned this yesterday, but again, when you start really looking at microorganisms, you can kind of understand where people that, kind of border on that kind of germophobic or germaphobes or whatever terminology for it, you understand where that comes from, because it can be quite overwhelming to, be exposed and see that the reality of that. And there are various ways to classify these. We're going to talk more about kind of their different forms here in a minute, and two specifically. But those are just kind of a general breakdown and characteristics of bacteria. We mentioned yesterday this kind of idea of taxonomy and classification. You can see here the amount or the breakdown of your bacteria versus your other, plants, fungi, animals, archaea, and you can see the great complexity and number of those actual microorganisms, of those actual bacteria. So in terms of number, they way outnumber any of the other groups. So as we look at their actual structure, and again, I think it's important that there's certain aspects of this that are going to be relevant as we begin to dive down into coffee fermentation. And, one of those, for instance, is the flagellum, the way that it facilitates movement for this microorganisms. And oftentimes, and I hear, people discuss often when we start talking about inoculations that, having a submerged fermentation or having water in that tank or in that system facilitates better movement of your microorganisms through that system. And there is some reality to that. Although just a reminder, we're talking about microscopic organisms. And so there is moisture that is there that they're able to utilize differently. But the reality is, yes, that flagellum is going to facilitate that microorganism's movement. And then you have these little pili, which are their kind of hair-like structures that allows them to actually attach to the surface of whatever it is, of different organisms, of of other bacteria, whatever it is that allows for that adhesion and for sticking to adhering to those different items. Then we have the cell wall, which gives that kind of rigid structure. So, while you do have that kind of unstructured nucleus and nucleus membrane, you do still have some of your cell walls. That is all about really protecting the plasma that's inside of that. So it does provide some degree of protection for the components inside of that. And also this capsule, you can see here this term capsule. It helps to prevent that bacteria from drying out, or even being kind of engulfed or swallowed by other microorganisms. And again, I don't want to get into the significant complexities of all of this, but more so just kind of presenting this from a general concept standpoint. The other thing to note is that the structure is, you see the cytoplasm, which is all the material in which these different ribosomes, which you can see kind of these different red dots here, where all of that genetic material is suspended. And again, so much of, we think about, I have to remember that the point of this bacteria is not to produce those metabolites that make our coffee taste delicious, but its main goal is all about sustaining its life. The ribosomes are structures that create proteins. And if you remember from our discussion yesterday, proteins were that really important chemical compound that's produced that's then broken down to feed certain types of metabolic pathways. So you remember that from yesterday. And then your nucleoid, which is your genetic material, your DNA. And so that's obviously an important part of the providing the blueprint, so to say, of how that bacteria will grow into more bacteria. So with bacteria, we turn our focus to different types of forms. And I only, again, a lot of this information is just laying the groundwork so that when we use these terms later, you're not hung up on what the meanings are or understanding those words. But we do see that bacteria does come in different shapes and sizes. And you will find these terminologies utilized in their naming or in their kind of classification. So the first one is the cocci, which are spherical in shape, roughly 1 to 2 microns. An example of this, which is one we often hear about in coffee, is Lactococcus lactis. And we'll talk more about some of the bacteria, and specifically lactic acid bacteria, which is in that category, as we dive into more discussions later on in this lesson. So cocci is one of the forms. The other forms is your bacilli. And bacilli you see here are more kind of rod-shaped, anywhere between 1 to 10 micron, and you have Lactobacillus plantarum as an example of your bacilli. And you can see the bacillus kind of as part of that naming. So you see where some of those forms and some of the names have interaction and connectivity together. So as we look at coffee, and just as a side note again, some of these studies are available for free online. So this one specifically is a study out of Australia. If you're interested in getting them, if you're interested in reading some, you can always use the author's name to search. And if you can't find anything, just drop me a note. I'm happy to flick it across to you. So that's no problem at all. But this specific study was interesting because they were able to identify different type of bacterial species. And there's a couple different ways they go about identifying microorganisms during these experiments. And so depending on whether it's a culture method or other types of methods, you can have some variations. However, an interesting thing that was noted in this specific study was that they found that your aerobic, so again, in the presence of oxygen, mesophilic bacteria, and if you'll just kind of put a note on mesophilic. I'm going to jump back to that at the end of our discussion today. But your aerobic mesophilic bacteria dominated this in this system, particularly they talk about a wet fermentation of coffee beans. We're talking about a submerged fermentation, meaning happening underwater. You have these different types of species in genera of bacteria, your Acetobacter, Lichenostic, Bacillus citrobacter. This citrobacter and Acetobacter are two that you'll very, very commonly find in a lot of studies. Oh, yeah, I can do that. I apologize about that. And you'll hear these found very frequently in different studies. And so yeah, that's something that you'll notice or that you might find quite commonly. Oops, sorry, I moved the screen a little bit. There we go. So what this study proved or showed, and what you can see here in the graph, is that bacteria grew throughout the fermentation, though you can see that there is different rates of growth depending on the different type or the different species. And you see that there is a period where some of them begin to die off. The different bacteria are utilizing the sugars that are in the mucilage as a food source, and they're producing different byproducts through that process. And one of those is lactic acid. Now, and I'll talk more about this here in a minute, but there are a couple different things that are happening during this process. We talked about the byproducts that are being produced. So there's all different kinds of metabolites that are being produced. But often these fermentations, not often, but they are increasing acid production, which decreases the pH. And that's a really significant activity that occurs. And there's a lot of research around how that acidification of the environment influences pectin removal or pectin breakdown and mucilage removal. So just kind of, I've got the next slide, we're going to dive into that a little bit deeper. In this study, it also showed that your Acetobacter and your Enterobacter were the most dominant aerobic mesophilic bacteria. And this study did, and I didn't note this, but just from reading it and studying it previously, I remember that there was a new species that they identified during this study that had not previously been identified in coffee post-harvest processing research. Now, that doesn't mean it didn't exist or it doesn't exist anywhere else, but again, it kind of speaks to that idea or notion of, does the uniqueness of the microbiota in a region or in an area, how does that play a role in the uniqueness of flavors and profiles that we see in coffee throughout the world? There's another study that identified over 168 different types of bacteria, and 44 of those that were responsible for degradation of the mucilage or decomposition of the mucilage. So we're definitely seeing and understanding that there is a complexity of the types of bacteria that we're seeing in coffee post-harvest processing. Now, one of the biggest challenges that we have is actually kind of parsing out and figuring out which ones are contributing to flavor, what types of flavor, how do we control that, how do we direct that? And that's kind of the world that we're stepping into and we're engaging more and more. And we'll discuss briefly when we get to connecting this with quality, with flavor. So as I mentioned, the one of the key activities that we find in coffee post-harvest processing is this breaking down of that mucilage. And most of your fruit fermentation, so that includes apples, it includes mango, other fruits, have what we call pectinolytic bacteria. Okay, and there's two examples of those here, Erwinia and Pseudomonas, which are both native microbes, not necessarily inoculated or anything, but native microbes that have these pectinolytic bacteria properties. Um, During these fermentations, there's three different enzymes that are produced, and how they're produced and the pathways are not necessarily as important. It's I think we'll just kind of keep it simple at this point, but they are this pectin lyase, polygalactouranase. I know you have to like practice saying all these big words. It's like a prerequisite for being able to teach it. No, I'm just kidding. Usually, I find that as long as you say it confidently, nobody questions it. Pectin methyl esterase. So those are the three enzymes that are produced by different by microorganisms that are all contributing to that pectin release. Now, there is a really interesting kind of debate that's going on in the scientific world, and that goes back to research, 20, 30 years, that basically looks at, is it bacterial activity that's actually breaking down the pectin and releasing it and allowing that mucilage to be washed free, or is it the acidification of the environment that's creating a situation where the mucilage can wash free? And one researcher, Avalon, had a really interesting study. He was looking at coffee fermentation in Mexico, and they looked at different types of bacteria and yeast, and they found that none of them showed strong pectinolytic activity, meaning the ability to release those enzymes that break down the pectin so that mucilage can be washed free. So their conclusion was that the mucilage degradation that was occurring was due to the decrease in the pH, which allowed for the decomposition of the mucilage in that kind of acidic environment. And I only mention that, so we ask the question, well, which is it? Is it the microbes? Is it the acid? What is it? I don't know that I wouldn't say that that's kind of main point. The main point here is understanding that, we talked yesterday about kind of some of those definitions of fermentation, right? And the idea that there is this sense of functional objective that is important in coffee fermentation. And so one of our questions becomes, well, if this functional removal of the mucilage is done by microbes, then are there microbes that are better for that or better positioned to do that, or is it just having a drop in pH? And why, kind of as I have this conversation in my head that I'll bring you along for the ride, then I ask the question, okay, well, if we're only wanting to use microbes to remove the mucilage, well, there's a way to do that with the machine that's probably more efficient and more consistent. However, if it's the acidification of the environment, that begins to step into a dangerous territory where we can begin to compromise quality if that extends too far. So I didn't mean to identify that this kind of discussion was unimportant. It's definitely significant and important, but we need to, one, if we first understand what are we trying to achieve in fermentation, is it the development of flavors? Is it just mucilage removal? Then we would change the approach that we would make. And as we begin to understand, these microbes being active and producing metabolites are going to lower the pH. But what would be an interesting study, and it would be very hard to control this, is to try to determine if we could acidify an environment without any of these types of microbes that are pectinolytic microbes. If we just acidify the environment, does that then allow for mucilage removal? Which is an interesting hypothesis, and I think we'll continue to see more and more studies being done. But the point is that during fruit fermentation, there are pectinolytic bacteria that are present that have been shown in other research to be responsible for decomposing mucilage, or is it just a drop in pH, acidification of the environment that allows for that decomposition to occur? Existentialist crisis here, right? I'm just kidding. So we're going to talk about three different types of bacteria, and I want to be clear that these are groups of bacteria, so they're kind of subgroups, I guess you could say. And the first is our acetic acid bacteria. And you'll often see this kind of written as AAB, acetic acid bacteria. Acetobacter are also another way that we describe this subgroup. Um, but they are most associated with acetaldehyde and ethyl acetate metabolites. Now, acetaldehyde has a strong fruity odor, and it's also known as ethanol, not ethanol, which there's a little bit of spelling difference, AL instead of OL, but it's not extremely significant. But it forms naturally in human body and plants and other places. But in really high concentrations, and this is where kind of within the wine industry and wine fermentation, acetic acid bacteria is actually considered more of a spoilage bacteria, because they will produce acetic acid. That's why they're called acetic acid bacteria. We like to kind of keep things simple. And that's, overproduction of acetic acid is going to be that, vinegar, and is going to create that intensity of those flavors in coffee, in the final cup. Now, ethyl acetate is a colorless liquid, typically has a sweet smell, but it's used in lots of different industries. In fact, some glues use ethyl acetate, nail polishers, but it's also used in the decaffeination process of tea and coffee. There's an ethyl acetate kind of type of decaffeination that you can use. But the thing to note about these acetic acid bacteria, and I apologize that this wasn't need to include this information here, is that they typically thrive in an oxygen rich environment, so an aerobic environment. And that's where, some the different, whether it's the characteristics or the flavor profiles that we see in natural processed coffees, or in a maybe a dry fermentation, meaning no water, more exposure to oxygen. You typically find these in your oxygen rich environments, your acetic acid bacteria. And I am tracking on the questions, so please don't hesitate to throw something over in the questions if you have anything. Then we get to our lactic acid bacteria. You can see again your lactobacillus, your bacilli. So you kind of see the structure, the shape of these bacteria microorganisms. They are going to ferment glucose into lactic acid. Apologies for the typo here. That bothers me. I'm going to fix that real quick. But they are going to ferment glucose into lactic acid. And we are going to talk about glycolysis, and I'm going to get there. But what they do is, through the process of glycolysis producing pyruvate, pyruvate is able to then be, through that production, lactic acid is created and allows for the oxidation of certain compounds that are then responsible for continuing on the glycolysis process. And I'll discuss more here in a minute. I got a little graphic I'll show you, I'll walk you through. But the interesting thing about lactic acid bacteria is this: they typically will grow, they all of them, they grow anaerobically, okay? Meaning that their metabolic pathways are all done without oxygen, okay? But interestingly enough, this lactic acid bacteria, most of your microorganisms that are either what we call, grow in the presence of oxygen, like your acetic acid bacteria, or with no oxygen, they don't always necessarily mix between the two environments in terms of their metabolism, except you do have a category that does, and we'll talk more about that here in a minute. But the interesting thing about lactic acid bacteria is they are what we call aerotolerant, which means that the way that they metabolize or create metabolites is without the use of oxygen. Okay, so it would be classified as an anaerobe. But it actually, even in the presence of oxygen, or when there is oxygen around, labs still maintain an anaerobic metabolic pathway. Okay, and that has to do with how their microorganism is structured. and the way that they produce the byproducts and gain energy. And that's the key here is so many of these processes, whether it's with oxygen, without oxygen, it's how the processes they use to create energy. And that's the key part of this discussion. Um, so and you know we've started to see a lot in coffee about these like lactic acid fermentations and what does that mean and what does that taste like and what's being produced. And yes, you do find that yogurt is a you know one of the products that relies on lactic acid fermentations, but so are pickles, so are many other food products. And so you know, it was kind of interesting. I think when lactic acid fermentation started kind of coming around, people were describing them as, oh, they're yogurty. They have this, and it was like this kind of connection to. I mean, I literally, we even had producers in Yunnan that were like throwing yogurt, you know, into their coffee fermentations as like a lactic acid bacteria fermentation or lactic acid fermentation, and it's not necessarily that straightforward. There are certain conditions that lactic acid bacteria thrive within, temperatures, etc. So, you know, earlier there were some people might not have been on, but Brian was sharing about how you know his reverse carbonic maceration coffee that they were selling. And so sometimes the allure of marketing things in a certain way might drive certain decisions of naming, but that's okay. We again, I'm not positioning myself as the word police. um, I do want to let you know I realize this now as well. This picture here has that kind of more rod shape, the bacilli type form. We do see that there are lactic acid bacteria that are like lactococcus we mentioned earlier that are more of your spherical shape. So it's not that all lactic acid bacteria are going to have that rod shape. It's just kind of an example of one of those. All right, let's talk about yeast. And I mentioned previously how um, there is, yeah, we'll get to that here in a minute. I mentioned previously how there's some microorganisms that will grow and thrive within different types of oxygen and different levels of oxygen saturation. And when we get to yeast, that becomes really an important consideration. So our yeast are typically single-celled fungi capable of fermenting organic, animal, or vegetable matter. So you know whichever that may be. Their shape is they're generally oval. You kind of see general size, and then you see you know a significant amount of species. The again just to remember, you know, we talk about yeast, molds, bacteria as if it's like one singular thing. We're talking about a host and thousands of different species that all have, you know, variations in their metabolic pathways and metabolites they're going to produce. You can see here that your fungi are classified as eukaryotes, and they are more closely related to our animals and plants in terms of their cellular structure and so forth. But they are both our yeast and molds are all going to be present or represented by this kind of fungi kingdom. So I want to mention briefly about this brewer's yeast or Saccharomyces cerevisiae, and you know it's here described as the yeast elephant in the room. Basically, any kind of discussion or research article, or you know, typically even outside of coffee, when you begin to talk about fermentation, you oftentimes will see this S. cerevisiae, Saccharomyces cerevisiae. And you can see this graphic here, this picture here represents the wide variety of different types of, or the complexity of the this organism, the genetic diversity. And you can see within different regions of the world, you have variations in even this kind of species of Saccharomyces cerevisiae. And we kind of explain it as a model organism, and the reason why is because of its stability, and the ability to not only, I mean, a lot of reasons why your industries are using yeast inoculants like your Lalaman, La Cafe, even other food and beverage companies use yeast is because they're very easy, they're comparatively easier to dehydrate and then to rehydrate. And so, whereas bacteria have significant larger amounts of limitations in your molds, potentially spoilage, but your yeast microorganisms have a lot greater flexibility. Um, the Most often, when fermentation is being discussed in other food and beverage industries, we're referencing Saccharomyces cerevisiae. Of course, there's lots of variations in that, but that's kind of what is kind of the workhorse, so to say, of fermentations. The in One of the aspects of, and I'm going to throw this term out there and I'll explain more of it here shortly, but yeast are known as what we call facultative anaerobes. Facultative anaerobes. And what this means, and again, I'll have that here shortly if you like. How do I spell that? Because I know I would be lost as well. Facultative anaerobes. The understanding is that depending on the environment that the yeast is living in, whether it's an oxygen rich or oxygen starved environment, they switch their metabolism. So you have certain organisms that can't thrive in oxygen, others that only thrive in oxygen, or you have some that can survive in both, but they switch their metabolism in order to adjust to the environment that they're in. And yeast, our particular significance is that they are a facultative anaerobe. So when they are in a oxygen-rich environment through aerobic respiration, which we talked about yesterday, they will produce energy. Whereas when they're in a no oxygen environment, they will switch to fermentation as a way to create energy, to create ATP. And again, I've got I got a cool little graph that'll show that here shortly. When we talk about their structure, we do see that they have a cell wall that is rigid. It gives it structure and it protects the plasm inside of the yeast microorganism. The membrane is a rather thin layer of phospholipids and proteins, and that's what controls movement in and out of the cell. Okay, so when we talk about those phospholipids and protein layers, bilayers, you see that that's kind of, for lack of a better term, the gates that provide movement in and out of the cell for various compounds. The vacuoles, which you see here, this kind of purple, dark gray, purple blob, is a vesicle inside the cytoplasm that is generally used for either storage or disposal. So we're going to find various compounds present there. The mitochondria is where we generate most of the cell's energy. And the mitochondria is going to be a very important focus of ours here in a minute when we begin talking about glycolysis. And then the nucleus contains the DNA. And you can see, again, yeast are eukaryotes. They have organelles. They have a very structured and rigid cell membrane, or nucleus membrane that protects the nucleus. And their ideal growing conditions. Optimal kind of cultural temperature is around 25 to 30 degrees Celsius. Apologize for those who, well, maybe not as many as, I forget the breakdown. But anyways, 25 to 30 degrees Celsius. And these yeast can be what we call psychrophilic, mesophilic, or thermophilic. And again, I'll define that here shortly for you, but essentially it has to do with the temperature range at which they are able to survive. Typically around 52 degrees Celsius is when we see the vast majority of yeast begin to die off, which yes is, not it's quite elevated in terms of temperature. But that's when we do find that they begin to die off, and they are resistant to low temperatures and freezing, which again is why they're kind of, many yeast are your kind of model organisms, because they are able to resist that type of environment, so kind of packaging and then rehydrating, etc., is a little bit easier. And they do tolerate a very wide pH range, from 2.4 all the way up to 8.6. So very versatile, very robust, and significant contributor to many food and beverage industries. Um, these are some of the common genera that we see in coffee processing. You see here like the wash process versus natural process. These are just a very, very, very few amount. One study in Brazil was showing over 140 different species of yeast just in coffee post-harvest processing. So you see our old Saccharomyces here. Natural process, again, slightly different because of the aerobic environment, and so the yeast that will thrive and proliferate in that. The other thing that can be a limiting factor to yeast development is other types of compounds that would develop. And so, you might be thinking, well, you just said that yeast was a facultative anaerobe, can exist both in aerobic and anaerobic environments. Why wouldn't there be more, yeast species in this natural process? Or why would some of them be there and not in the wash process? And a lot of that has to do with other microorganisms out competing the yeast that are being kept at bay. And that's one of the things differences that we find, or one of the things that impacts. And as I said, many other species that are involved, and we'll talk more, as I said here, and provide some more information on some different species and linking that to flavor. All right, and then we're going to talk about molds as we wrap up this lesson, talking about structure, classification, and then what their role is in post-harvest processing. There was a question about, if you send me a question in a direct message, I'll just kind of keep it anonymous who asked, just because I want to protect your privacy, but I will kind of read the question. Oh, all right, Ibrahim. Sorry, I didn't know if you were concerned about sharing. Okay, my bad. So like bacteria, yeast present in coffee naturally in the post-harvest, yes. So what we do understand, what we do know, is that both bacteria, yeast, and molds are all naturally present. What we call them are native microbes, so to say. And yes, the development or growth of microorganisms throughout the world, through trade, through different activities, yes, that does happen. But we would kind of call them our wild. Sometimes people use native, some people use wild. But yes, they are present in coffee naturally in the post-harvest process. And they come through every means, from the people that harvest, to the wind, to dirt, to other animals that are in the area, to pests. They're attached to everything. I showed you that's one of the reasons why I pointed to those pili, because those bacteria will adhere in all kinds of different places, and then they kind of come along for the ride. And so, yes, they are naturally present. Buyers who make Instagram posts, yes, Anna, that is correct. When you travel to the coffee farm, you are bringing your own microbiota with you. That is correct. And in fact, an interesting, when I was in Hawaii, so Hawaii, for a long time, coffee leaf rust is a mold that impacts coffee production throughout the world. And for a long time, Hawaii didn't have any coffee leaf rust on the islands. And in recent years, it's developed, and they're putting a lot of research behind trying to curb its growth, because you see in some parts, like Latin America, like Ceylon, Sri Lanka back in the 1700s, it can completely decimate the production in that region. And so they actually, one of the farms that I went to where we did a course, when you pull up to the gate, they ask you to spray like your shoes, your tires, any kind of things that touch the surface of where you're walking, with a sterilizer to try to limit the kind of proliferation of those microorganisms. So maybe you'll have to Anna start being in like a bubble when you go to like coffee farms. It'll be these pictures of like Anna in a bubble suit. Anyways, bad joke. Okay, we'll move on. So molds. What are molds? They are essentially your filamentous fungi is the technical kind of description of them. They eat externally. Okay, what that means is that they're going to secrete enzymes that are going to break down the compounds externally that they're going to consume. So there's a huge amount of work that's done by molds in decomposition. And they are what we call osmotrophic, which is this idea that they secrete enzymes that break down compounds that then they can consume. It's called osmotrophic. But the we hear a lot about molds, both at a level of storage, food safety, etc. Um, but they are not. So we hear a lot about them in coffee production. Alex, lost connection. Kindly, could you please repeat the last part at the farm? Oh, I was just saying that like the farm that I was at, they had you spraying sterilizer on your shoes and feet and stuff in order to limit the growth, the spread of the spores. You know that you wouldn't be a carrier into a farm because they were trying to limit the growth of this coffee leaf rust. That was all. It was a little bit of a tangent. Sorry. So some of the common molds that we find, they are widespread. They can grow in diverse conditions. But here's a significant part of molds is that they require water and oxygen. So they are what we call obligate aerobes, which means that they must thrive, and they only thrive where there is water and there is oxygen. So, and that just to be clear, that water and oxygen can be in different forms, and I'll talk more about that here in a moment. But when we have, and that's why, like when we talk about drying coffee, or we talk about natural processed coffee, or we talk about others, where there is water and oxygen, there is the potential for mold development. And I recall we were working in Yunnan, we had installed a bunch of greenhouses, parabolic dryers. They kind of go by different names, but basically this kind of covered structure that we were drying coffee in. We filled the whole thing. It was about, I don't know, 15 tons or so of coffee fruit that we put inside this thing. And I remember the night before, I said, please don't close the doors when you leave. Leave the doors open so that, there's good airflow and stuff through. And I come back the next day and the doors are closed, and it was like a sauna inside. And within, I mean, that was only 8 hours, even maybe 10 hours the most. You already could start to see mold start developing on the surface of the coffee because high water, high oxygen, proliferation of molds. There was no air movement to kind of move that water and oxygen along. So just something to remember when we think about mold. And we say, well, is that good or bad? Well, we're finding now some mold is good, some mold is bad. Some, the kind of koji fermentation. I don't know if you've followed anything of that recently, but that's becoming something that people are pursuing. So you often see this term hyphae that is used with in relation to molds, and it's basically the kind of structure, long branching structure that goes out and is a part of the spread or the growth of the mold. And oftentimes it's kind of collectively referred to or called the mycelium. And I have a great friend. She does processes and works in coffee in Myanmar. And they've been doing some really interesting stuff with mold development and mold growth, both at a composting, but also specifically with processing coffee, with processing naturals. And she sent me a picture one time, and you could see the mycelium kind of growing and spreading and stuff. But and the cell structure of molds is like other eukaryotes, just like yeast. So I'm not going to dive and go through that whole thing again, but their structure is similar to that of yeast. But a key function and a key difference and importance with molds is that they reproduce through spores. And if you ever taken like a sensory class or a green coffee class that talks about the fungus defect in coffee, I'll be honest with you, I rarely ever come across it because it's one of those things where when you find it, you have to isolate it and pull it apart because it'll spread. Because there's those spores present and it'll spread because that's how molds and funguses develop and grow. Similar to, the house that I live in here, in the US, we have these HOAs, homeowners association. And my dad was telling the story about how he would get this note every now and then because they want you to spray the side of your house. We have really humid conditions here in Houston, like 90, 100% humidity. It's unreal. And during the summer months and kind of with the rains, you'll get this mold that will start to grow on the side panels of your house. And they actually require people to take a solution. It can be a chemical, it can be a natural solution, whatever it is, and spray it because they're not necessarily just concerned about your house, but they're concerned about it spreading to other people's houses. So it's like a way of helping to monitor this community spread of this mold because it spreads by spores. And that's something that those spores can blow through the air, and even if you're interested in any of the history development of the coffee leaf rust, um, Cooker, I can't remember his name. Maybe somebody knows it, but there's an author, Stuart McCook, I believe is his name. That could be wrong. Don't quote me. But he's written a book on the history of coffee leaf rust and kind of talks about how potentially wind currents took the spores of coffee leaf rust to different parts of the world. Interesting historical progression. Temperature range. Give me just a second, Ibrahim. I am going to cover that here in like a few slides. You're totally providing me the great segue to move through this. So yeast versus molds. Yeast are typically single-celled, whereas molds are going to be multicellular. You won't find those mycelium in yeast like you will in molds. Yeast mostly produced through mitosis or through budding, so it's splitting, and that's how they reproduce. Whereas molds produce through spores. Yeast can thrive in aerobic or anaerobic conditions, whereas molds are specifically only aerobic conditions. And the other key aspect of those is that yeast are going to gain energy, that ATP, through fermentation, whereas molds are going to gain that by breaking down those organic compounds externally and consuming those compounds. So that's some of the key differences, and the way that different metabolites are produced by these different microorganisms are key because of not only the conditions, but even how they go about doing it. The enzyme that is excreted by these molds can play a role in the environment that it creates. The last thing I'll mention here is this kind of understanding of mesotoxins, which is something that you've probably heard of as you talk about coffee, and specifically ochratoxin A. It is something that is monitored, and even some countries have regulations on testing coffee samples as they come in because it is something that we can see in coffee as a defect and is harmful for human consumption. And interestingly, I used to hear, oh, well, it's not a big deal, it's roasted away. Well, actually, some of the research is showing that it's not fully roasted away. So it is something that poses significant risk. But we do see that if we are drying coffee to below 12%, that we will see a significant reduction in the probability of this type of mold development. So drying below 12%. And why 12%? And it's because at 12%, that's when the water activity threshold is at a reduced level to where we're not concerned about mold development and mold growth. All right, last thing I'm going to talk about, and then we'll jump into some discussion of the fermentation process, is their growing conditions. I don't know how you feel about these cute little illustrations, but I have small children, so humor me. So the first one, and this is where I told you I was going to show you this illustration, and I'm already making notes in future iterations. I'm going to throw this up to the top so I don't have to keep saying, I'll explain this in a minute. But we understand that the growing conditions for different microorganisms is heavily connected to the levels of oxygen. And you see here 4 different classifications of of how we can understand microorganisms. You have an obligate aerobe, which means that it needs oxygen. Okay, so it utilizes oxygen in its development of ATP, in its creation of energy, and its sustaining life. And you can see here this kind of first test tube showing those obligate aerobes living at the surface of solution or water or system or whatever it is. So they're hanging out where there is a large amount of oxygen because they need a significant amount of oxygen in order to survive. Whereas you have an obligate anaerobe, which is basically that oxygen is toxic to them. Okay, so you find it in like stomachs, like stomach acid, or swamps, or other places where you have a significant reduction in oxygen, and those are obligate anaerobes. And because of that, what they use to create energy to sustain life is going to vary. And this is where, if you remember on the first day, which was yesterday, I talked like that was such a long time ago, right? Yesterday we talked about those extremophiles, those microorganisms that are living and thriving in those extreme conditions. Many of these are going to be your obligate anaerobes. Then we have our facultative anaerobes, I just mentioned, which yeast are a significant part, or one of those significants. And depending on the degree of oxygen or the level of oxygen, they switch their metabolism to adjust. So if there's oxygen, they're utilizing aerobic cellular respiration, whereas if there is no oxygen, they're switching to anaerobic cellular respiration, which oftentimes is fermentation. And then we have the fourth classification, which is aerotolerant. So these microorganisms do not use oxygen. So whether there's oxygen or not present, they will not utilize that oxygen. And so their metabolic pathways and metabolites are going to be impacted. Okay, so that's oxygen levels. Then we have your question here, Ibrahim, about growth temperatures. Okay, so we can understand and we can categorize different microorganisms by the temperature in which they grow within. Psychrophilic being below 20 degrees Celsius, mesophilic, which is in that kind of 10 to 45 degrees Celsius range, which is typically speaking what we find for most coffee fermentations. Okay, now I'm not talking about your cooler storage or your freezing coffee fruit or other things. So not talking about those situations, but under normal fermentations, you're looking at mesophilic being the type of microbes. And that was why earlier I mentioned the aerobic mesophilic bacteria as being the dominating species. Well, kind of makes sense, right? Because those are the environmental conditions that are existing in that situation. And then we have our thermophilic, which is above 45 degrees Celsius. And you can also get hyperthermophilic, which can even go above that. Philic, something loving. It's a really good question. Yeah, I think philo is kind of to love. I think that's right, Tristan. Don't quote me on that, but I think you are. So it's like loving that kind of temperature. Yeah, language is so cool. And then the third aspect, so we have oxygen, we have temperature, and then we have pH. And everybody, I'm sure, would be familiar with your pH scale, kind of ranging from 0 to 14. The smaller the number, the more acidic. The larger the number, the more alkaline, the more basic. And you have microorganisms that will thrive within different types of environments. So you have your acidophiles, which are going to thrive within the kind of 3 pH range, give or take, neutrophiles and alkylophiles. Most of your bacteria are neutrophiles. So you can kind of see in that kind of 5.5 to 8.5 range. Now, what's interesting is you do have and this is where, as we think about the system of microbes in a coffee fermentation, we can see how certain compounds begin producing byproducts and begin dominating, and then maybe as the pH level drops, you have other microbes like your yeast begin to take over. So when we think about how do we control, like once we understand, okay, this yeast species produces these kind of metabolites that we want to encourage, how can we control that? Okay, well, can we limit the pH? Can we change the temperature? Can we do different things to minimize other interactions? But the challenge with coffee, and this is where you think about a winemaker and a winery, what's fermenting wine versus a coffee washing station or wet mill, in terms of the sterilization, the cleanliness, etc. I'm not saying one's better than the other, just identifying the differences. You can see how even if we were able to understand, okay, this one species thrives at this pH and this temperature, and we say, okay, we're going to put the coffee at that state and that condition, and they're going to take over and go, but that's assuming that nothing else thrives at that condition, and then that's the type of scenario. So that's where the properties and inoculation, and we'll talk more about that shortly or in a couple of days. That's where that becomes something we can consider and pursue. So microbes are diverse. Now, what do they do? So I'm going to jump over. I'm watching our time. Like I said, this one was going to go a little bit lengthy today, but we're going to jump into talking about the actual fermentation process. So, yeah, Anna, your point, monitor your fermentation tank to know what's going on. It is true, but the real challenge is that if we my experience has been in coffee that there's times when I can do the exact same thing day to day, exact same way, monitoring at the exact same temperatures and everything, and you can have variations in your outcomes. And a lot of it has to be with the condition of your raw material, the influence of certain microbes, humidity levels of the environment, things that really are going to be difficult to control. Now, I can do many things to try to mitigate that. I can soak the coffee fruit to try to remove the microbial load. I can cover my fermentation tanks to try to reduce environmental impact, etc. But it's amazing to me as we study more and more about fermentation and we learn more about post-harvest processing, just how incredible it is that we're able to see such diversity of flavors and flavor profiles with such little quote unquote control of the process. And whether that's a good thing or bad thing or something we'll see more of or less of, I don't know, but it's just amazing to me. And that I know that many of you work on the consuming side of the industry as baristas and roasters. And I'll just be honest, when I drink a great cup of coffee, though I am grateful for the barista and I'm grateful for the roaster, my first thought is always, man, that producer nailed that, and that's kind of where my mind goes. And again, not to diminish, I see people leaving. No, I'm just kidding. Not to diminish at all the roaster, the barista who served it and brewed it and roasted it, but yes, it is why a significant starting place is monitoring and beginning to understand what's happening, and then connecting that to a sensory outcome. And I always really that's my thing is taste. Such an important part of our investigation. So we're going to discuss about 4 main phases. So in this lesson, we're going to look at fermentation phases. We're going to talk about glycolysis, and then we'll talk, if we have time, about some different fermentation pathways. And I already kind of see that I'm probably going to run out of time, but that's okay because we have 3 more days together, so no worries. So we look at 4 different phases of fermentation, and the first one is your lag phase, okay? Now, the key part of the lag phase is adaptation. Okay, so what's happening is you have microbes that are being introduced into an environment and they're adapting. Now, this could take 1 hour or it could take several days. It's not necessarily a straight across the board when that lag phase is over. During this period, you have little to no cell division, but cells are still active. So they're adjusting to the temperature, what type of compounds are around them. They're basically, again, we anthropomorphize a lot, but they're getting used to their new home. They're checking things out. The length of this lag phase is going to be influenced by the type of species it is, what they're being introduced into, even what their initial size is. Do they need to spend time kind of working to outcompete the other microorganisms around them? And temperature is another big one. Higher temperatures tend to reduce the time of lag phase. And a question that we often get in discussing this is, well, when do I know when my lag phase is over? And really, it's because of different activity. When you start to see that bubbling and production and actual byproducts that are being produced through that process, whether that's an aromatic, whether that's actual CO2 production, that's when we know that lag phase, that we're moving into the next phase, which is the log phase or the exponential phase. And that's where we see significant growth of our microbial population. And you can see here how certain microorganisms, typically our yeast and our bacteria, how they grow. It's in this exponential type of growth rate. The progression of this will continue for as long as there is conditions that exist for those to grow. And as long as there is substrate or food available to be consumed, and as long as nothing has outcompeted it, that microorganisms will continue to grow through the log phase. And log relates to the exponential growth in a mathematical log, but that's not extremely significant where the term came from. That's just what it's called. And then once we see that progression of growth or microorganisms, we then get to what's called the stationary phase. And to be clear, during the stationary phase, you still have production that's occurring. So you do have, and this note here particularly is about secondary metabolites, and that's an important thing to consider because, as I mentioned, it's yesterday, it's not like your microbes line up. I always use the illustration of like the water fountain, which I guess is now like a thing of the past. But I remember in elementary school, we all lined up after recess at the water fountain, and everybody took their turn at the water fountain. Microbes are not like that. They're not lining up at the water fountain to take their turns drinking, and consuming compounds. One, they're in a race to trying to outcompete each other, but then also certain things that are being produced, other microorganisms are utilizing and producing secondary metabolites. So it's not this kind of linear, very organized progression, at least not as it currently stands in coffee, in kind of classic or traditional coffee fermentations. And one thing we find during the stationary phase is that the amount of organisms that are growing and being developed is equal to those that are dying and those that are ending their life. And so that's why you kind of have this stationary, you don't have any growth in terms of their numbers. And then the last one is decline or death phase, where you have this exponential decrease and cells undergo what's called lysis, which is basically where the membrane breaks down and they begin to decline in number. Interestingly, you hear people use the expression, my fermentation stalled out, or it's stalled, it's the fermentation stalled. So it could be that this happens because the microorganisms are dying, but more often than not, it's because in this stationary phase, the substrate or the food that they're consuming becomes all used up. And so the growth that you have of microorganisms is typically through those secondary metabolites. But again, those begin to run out, and then that's when you see the death phase. Though I guess, the main way if your fermentation is stalled out in the death phase or decline phase or stationary phase is if you were to add substrate to it, does it continue, does it take back off and continue to grow, would be a key indicator. All right. This is where we are going to now jump into, I just want to stop from here. Are there any questions? We've covered a lot. Microorganisms, bacteria, yeast, fungi, talked about fermentation phases. I forgot that I have to take a breath and make sure there's no questions. Apologies about that. What do you mean by adding substrate? Oh, great question. Thank you for asking that, Nico. So substrate, we use that term to basically describe anything that the microorganisms eat. So anything that they consume. So, that oftentimes in coffee we're talking about that mucilage, but really it's any type of, yesterday and we talked about carbohydrates, proteins, fatty acids, nucleic acids, all of those become substrate, become compounds that can be consumed by microorganisms. And so really that's what I mean when I use the term substrate. I mean essentially food. And depending on the type of microorganism that exists will dictate the type of compounds that they might consume. So, for example, the simple sugar of glucose is used in that process of glycolysis, and that glucose compound, which is a simple sugar, is something that um, you know uh, is essential. And if we have let's say more complex sugars, it's going to take time for those complex sugars to be broken down into simple sugars in order to allow for those to be available for fermentation or available to microorganisms to consume. I was going to ask something about that. Is it visual or is it something you have to measure to see if the process continues? Usually, so typically during a fermentation, you have carbon dioxide that's released, and you have heat, you have aromas as well. So anytime, like when we talk about, even when I do the Q processing level 2 course, and we do our hands-on processing, one of the things I always encourage people is to smell, because those processes of fermentation are going to create volatiles. And oftentimes, if we can connect those volatiles to actual results, sensory results, like when I smelled this, the outcome was positive. When I smelled that, the outcome was negative. That's a really good way to kind of combine other indicators, pH, temperature, etc. And so one of the indicators we could use would be even aromas. The volatiles that are being produced can be an indication that there is continual fermentation happening. But also, you generally find the activity of bubbling or effervescence as kind of an indication of, uh, you know, whether we added something back in, and there's continual fermentation that's going on, continual activity. Did you mean that we may add some additional food for microorganisms? Yes. Do you measure the pH going back up because you added food again? It depends on what it is. So if you were to add water, for example, absolutely. pH is going to jump back up because you're going to kind of introduce a higher pH to that environment. In terms of if the pH is going to adjust, it depends on the solubility of that substrate, so whether it can be dissolved by water or not, and the amount of water that's even present. So you probably would see some impact to pH, but I would imagine you're going to see continued decrease because you're going to have more acid production, because you're going to have more activity by your microorganisms. Is there any unwanted byproducts? Yes. And what are examples of secondary metabolites? Are they all wanted and contributes to flavor? It's a great question. If you'll allow me, I'm going to put I'm going to copy and paste that one and throw that into our a later lesson, because we're going to make those connections between some of the positive byproducts and flavor characteristics. But the answer is yes. Specifically, when we were talking about acetic acid bacteria, right? Producing vinegar, producing acetic acid. And I'm sorry, I should say producing acetic acid, not vinegar, but producing acetic acid. And that is a characteristic traditionally that we find in coffee that is attributed to a negative characteristic in coffee. And we're typically pointing to an overproduction of acetic acid. Now, I will say, you are finding, and this links a lot to growing consumption and new markets that are entering into, kind of the consumption market, consuming coffee. Their domestic consumption is growing. We're finding that some of those coffees with more, acetic acid or higher acidity are being prized or being valued higher than other markets. And for one, when we think about quality, right, and we could really get into a long discussion on what is quality, right? When we look at a strict value sense, basically the end consumer is the one that really connects what they prefer, what they like with value, what they're willing to pay for something. And we can tell people all day long, this is good quality, this is bad quality, but if people aren't willing to pay for it, is it really good? I mean, again, I don't want to get into too many of these like existential crisis, type of questions, but the I would say, and I teach the Q Arabica program. I'm involved with other types of sensory programs. I would say that washed Arabica coffee, I think in general, we have a really good sense of what the consumer identifies as high quality and low quality. And we have a pretty good consensus and calibration on that. Whereas experimental, extended fermentation, even naturals to some degree, there's not always a huge consensus on what is high quality and what is low quality. And honestly, I don't think that's bad. I actually think it's great that we have that diversity and we have that complexity. And so, what I think our job is as both processors, as sensory individuals, is to understand what's happening, to be able to articulate the type of characteristics, the sensory profiles of that, and then learn from different cultures and different experiences what is considered higher quality and what's considered lower quality. Because I mean, just as a very, very kind of condensed example, I mean, when I go to our Q instructor calibration events and we throw some naturals on the table and honey process and extended fermentation coffees, you'll even find within our group some mix on what's considered high quality and what's considered low quality. How sharp of acidity is, over fermented or fermented too long versus what's, preferable and what's, and then you talk about how I'm going to use that, how that coffee is used and how it's served and all of that. I mean, there's huge complexities to it. And to me, I'm okay with there not being a answer to that question. I think it's actually something that really leaves it to the hand of producers to be innovative, and then the consumers and roasters to be able to really offer a unique and different product to their consumers. And so I get from the producer side, it's frustrating because you're like, I just want to know how to produce really good coffee and sell it for a price that I can like provide for my family. Right? Like I totally get that aspect of it. And I think that's important. And that's where that kind of communication loop throughout the industry is so important. We're feeding back that information. Brian, not to continue to pick on you, but your reverse carbonic maceration process. Like if that was a really good product and sold really well, man, let's get that conversation back to the farm to say, you know what, that went really well. Let's look at offering that again, or maybe we expand it differently. And that communication loop really begins to strengthen and equip producers to extract more value out of the kind of end value of coffee as a product. Sorry, we're getting way beyond fermentation here. I actually tomorrow I'm doing a presentation for Vanderbilt University has the Wandry. They do this design equity lab, and they're doing a coffee equity challenge. And I'm speaking on economic sustainability in coffee, which is so that kind of value discussion really begins to play into. Does the length of each phase depend on the environment microbes been introduced to? Absolutely, 100%. Not only does it depend on the environment they've been introduced to, but even their characteristics as, are they, are they being introduced at a higher temperature, but they like lower temperature, and vice versa. Are they being introduced at a low pH or high pH, but they enjoy low pH? So absolutely, it is going to influence. Maybe I missed this. Can you give an example of substrates? Sure. So one example is the mucilage. The actual mucilage. So when you pulp coffee fruit and you throw parchment coffee with mucilage intact into a fermentation tank, that mucilage is substrate. It's consists of proteins, amino acids, carbohydrates, all of these other compounds that are available to be consumed. And that's what we mean by substrate. I also, some other interesting, you mentioned, Brian, maybe it was you as well, banana fermentation. When people are adding bananas into a fermentation tank, or mangoes, or fill in the blank, all of that becomes substrate. All of those materials are made up of compounds that can be consumed by microorganisms. And I don't want to touch yet. Like, is it good or is it bad? There's a host of other questions that we need to answer before that, or understandings. But that's what we mean by substrate, essentially food that microorganisms are consuming. Great questions. Thank you for those. The last thing we're going to talk about today is glycolysis, and then we'll wrap for today. So yesterday we started talking about this idea of fermentation as cellular respiration. Okay, I'm not going to go into depth repeating that, and we've already discussed that at length. But we understand that there are these processes of aerobic respiration versus anaerobic respiration and fermentation. What are the nutrients? I just mentioned this. We have sugars or carbohydrates, but we also have amino acids and fatty acids. And that was kind of that breakdown that I showed you, where I, in my most beautiful penmanship that I could muster, wrote out the different types of compounds. All living cells must carry out respiration. Okay, so all living cells have to carry out respiration in order to produce ATP. Okay, and that, again, that ATP, adenosine triphosphate, not extremely important that we know what that means, but just that we understand ATP as being that energy that's being produced that sustains life. And what are those metabolic reactions and where do they occur? Depends on the type of cell and the availability of oxygen. Okay, so let's talk about glycolysis. Glycolysis is the very first step in cellular respiration. Okay, and when we're talking about the organelles and the different structures and stuff, the cytoplasm, that is where this occurs. And it is going to break down glucose. You can see glucose. Actually, you can't because my cursor is not showing up on the screen, but you can see it there at the top of the circle. Glucose being broken down into pyruvate and ATP. Okay, so you can see in this illustration that one molecule of glucose is being broken down. If you remember, that is a catabolic process. So catabolic breaking down, anabolic building up. Catabolic process where glucose is broken down into pyruvate and produces 2 ATP. Okay, so that is the result of glycolysis. Now that process does use, and it's important to note here, this NAD plus, and it ends up in producing NADH. And it's not specifically important what that is, but it's a very important process to know that this process of glycolysis utilizes this compound. So it utilizes NAD plus and it produces NADH. So essentially what the point we're making is that it requires that NAD positive. From this point of glycolysis, so we have pyruvate and we have 2 ATP molecules. Now, what happens in an aerobic respiration? So this is remember yesterday when I showed you the metabolic pathways and there was like a circle and a line and a circle and then a line. This is the next, point on this progression. When aerobic respiration occurs, so that's when there is oxygen is present, oxygen is available to be used, that pyruvate then is utilized in what's called the TCA cycle, or it's also called the citric acid cycle or the Krebs cycle, but it's a very fundamental biochemical process within all living things. When there is oxygen present, that TCA cycle occurs and produces 34 molecules of ATP, as well as producing CO2, water, and other waste byproducts. But the key here is that aerobic respiration utilizes the pyruvate from glycolysis to produce 34 molecules of ATP. Now, when you add that to the 2 that were already produced through glycolysis, you have 32 molecules of ATP that are generated through this process, through aerobic respiration. Okay. Now I know you're all going to go to like your dinner parties this weekend and use this stuff and impress all your friends. So but why is that important? Why is it that 36 ATP is so important? Well, when we contrast that with fermentation, Okay, so that was an example of aerobic respiration. This is an example of anaerobic respiration, otherwise, or as we understand as, or in this instance, we're talking about fermentation. So what happens in fermentation? Fermentation takes the pyruvate, it utilizes this pyruvate, and it breaks it down through a catabolic process. That breaking down of pyruvate creates various byproducts, various metabolites, depending on the microorganism responsible, or what that microorganism is that's utilizing that pyruvate. And it utilizes the NADH from glycolysis. So it takes the NADH and the pyruvate, and it produces byproducts. In this example, it's using lactic acid fermentation. That's why lactate is there. It's not always lactate, just to be clear. That's just one of the processes. But through that process, it produces NAD plus that then goes back to be used for glycolysis. Okay, so remember, all of this whole fermentation is all about producing energy, producing ATP. If you have an organism that produces 2 ATP through glycolysis, how do you continue that wheel of production of ATP? You utilize fermentation. You utilize the pyruvate and the NADH from glycolysis to produce various byproducts and other types of compounds, depending on the metabolic pathway, depending on the microorganism, but you produce NAD plus that then is fed back into glycolysis, and the cycle continues and continues as long as there's glucose. Or I do want to note here that we're talking about glycolysis and glucose. You can, and specifically glycolysis as being rooted in glucose, but you can also find that other compounds are utilized to produce ATP. Okay. Great question, Santiago. And you know what? I knew you were going to ask me that. And I looked it up yesterday and I had it because sometimes, in my brain, there's only kind of like a certain amount of space for certain information, and the exact breakdown. So it's Nico. Here, I'm just going to copy this over here. I'm not even going to try to embarrass myself by pronouncing this. Oop, let's see, everyone. There we go. And basically, NAD positive is nicotinamide adenine dinucleotide. I didn't butcher that too bad. But when it gives its hydrogen ion, which the H is just a hydrogen ion, when it donates that hydrogen ion, it becomes a proton receptor, and that's the positive. So when it donates the hydrogen ion, it takes on a proton, which then creates NAD plus. When NAD plus is utilized in glycolysis, it receives a hydrogen ion, making it NADH. You're very welcome, Santiago. No problem. It was funny last night as I was doing some review for this. I was like, you know what? I should write out what NADH is. And I was like, nah, I won't do that. So I learned. It's like fermentation fuels a glycolysis process produces energy. That is exactly correct. That is exactly correct. And why we talk about this is because, okay, well then what's the difference between fermentation? Is all anaerobic respiration fermentation? No, because some anaerobic respiration utilizes different. There's no glucose, there's no glycolysis, and so it utilizes different compounds and produces different byproducts. But the key function of fermentation is to allow glycolysis to continue in an environment where there is no oxygen, or rather when oxygen is not available. Please let me change that. When there's no oxygen available, because as we discussed previously, there's different microbes that exist and can thrive within different levels, different environmental conditions, different degrees of oxygen. So when you have, that's why, and I'm going to jump back over to that slide here because I think it's on the previous one. Nope, just kidding. It's on this one. I'm going to throw that back up here. When we have a obligate aerobe, okay, so they're only thriving in oxygen environments. So an obligate aerobe is not going to utilize fermentation to produce ATP. Well, why not? Well, because look at it. They can produce 36 ATP, and not like they sit down and kind of run the decision matrix and decide which they're going to do. But you can understand how aerobic respiration is a way more efficient way to produce ATP as compared to fermentation. Through aerobic respiration, they're producing 36 ATP molecules, whereas in fermentation, we're only producing 2. Now, there are other byproducts that are produced, yes, but so are there in aerobic respiration. Whereas, and so you take like a facultative anaerobe, right? So they can switch, and you can understand why when there's oxygen present and available, let's use oxygen, way more efficient. Okay, I'm in an environment with no oxygen, I can switch my metabolism to fermentation. Aerotolerant is an interesting one because whether or not there's oxygen present or not, they're still utilizing fermentation and anaerobic respiration, anaerobic cellular respiration to produce ATP. So, again, I said the note about impressing your friends at a dinner party because you're kind of like, okay, well, like, why is this significant? Why is this important? And really, it ties to the definition of what we're actually calling fermentation, but it also helps us understand how we can better control the process. When we begin to understand that this process of glycolysis is essential to producing pyruvate, which is utilized in fermentation, we can better understand how we can control and monitor and intentionally ferment as we tie this to different types of sensory or quality types of outcomes. So I know we're getting close, about 10 minutes before we need to wrap. I am going to halt here. I'm right in the middle. We'll pick up tomorrow talking about different fermentation pathways, so like alcoholic, lactic, malolactic, etc. And then we'll talk a little bit about some different volatiles that we find, so your esters, aldehydes, and ketones. And then we'll kind of continue to progress through different materials. Are there any questions? I know we stopped and we paused for some questions, but any questions, thoughts before we wrap today? No? Okay, that's cool. I know you're like processing it all, which is totally fine.
I have a question.
Yeah, go ahead.
During the aerobic respiration, aerobic cycle on the left, do we use NAD plus for the glycolysis, something like that? I can pronounce.
That's all right. That's all right. So, what is during glycolysis? We produce pyruvate. NADH is also one of the byproducts. Pyruvate and NADH are used in that. Oh, I'm sorry. I'm realizing I'm pointing to things in the screen. It's not. Sorry about that. But on the left side there, where the little purple blob is, pyruvate and NADH are used in the TCA cycle to produce different byproducts, and NADH is a byproduct of that process. Now, how does that NADH then donate its hydrogen ion to become NAD plus? It's through the interaction, through the interaction with its environment, as well as the compounds that are in the system, in that system. So it's not as there are definitely more byproducts that are produced in this aerobic respiration than there is fermentation. I don't know if that answers your question or not. But you do have NADH as a byproduct of aerobic respiration. And then all we have to do, not that it's a small thing, but all we do is that NADH molecule donates its hydrogen ion basically to something that has higher polarity and then becomes NAD positive. How many molecules of ATP are produced in anaerobic respiration? Anaerobic respiration in terms of fermentation that we have here, 2. So you can see in this graphic here on the screen, glycolysis is going to produce those 2 ATP. All fermentation is going to do is allow for glycolysis to happen again to produce 2 more ATP, in addition to other byproducts, of course. But in terms of sustaining life and creating energy for the cell, that's the important part. For us, the important part, I mean, it is important that the microorganisms are able to survive and thrive because we want them to produce more. But the important part that we focus on, in addition to microorganisms sustaining their life, is the byproducts that are produced, those metabolites that are produced. Any other questions? I really appreciate you guys throwing questions out there. Doing a great job. I know, you know, trying to take in all this stuff and get notes. Like I said, I'm really sorry that we don't have a workbook. I've lobbied that hard because I know it's a lot of information and so much of it is probably things that you're hearing for the first time, and some of you are hearing in a different language, and so I totally respect and I apologize for that. Can we consider glycolysis as part of the catabolic process? Absolutely. So again, catabolic is any metabolic pathway where breaking down compounds. So glycolysis is taking that glucose, breaking it down into the subsequent compounds. So 100% it's a catabolic pathway.
Okay, so you said NADH is one of the byproducts of glycolysis, uh, glycolysis. And so, what about NAD plus? I didn't get it in the aerobic respiration.
So
this part here?
Absolutely. So in order for NAD plus, I'm sorry, in order for glycolysis to occur, we need glucose and we need NAD plus. Okay, those are the two of the key components to glycolysis. During the production of glycolysis, we have 2-ATP, pyruvate, and NADH. And what's happening in this process is the term already went up. I'm just going to use NAD because it's a lot easier to say, is accepting a hydrogen ion, okay, that's been created through this breakdown of glucose. And when you look at the actual chemical formula for glucose, you see that hydrogen is one of those compounds. And so that's where the hydrogen comes from. In aerobic respiration, one of the byproducts of this TCA cycle here on the left is NADH. That hydrogen is then donated. It's given away to another compound with higher polarity than the NAD. We talk about bond strength. So the bond that's holding that hydrogen to the NAD compound, there's another compound, whatever it is, that is higher amount, that has a greater polarity, that pulls that away, and the NAD donates that ion. It's what we talk about in chemicals. It's not like it, anyways, it donates the hydrogen to another compound, make it NAD plus.
So
and then that's utilized in glycolysis.
During the aerobic respiration, again, we need NAD plus, where do we get it? Again, in the aerobic respiration, we have fermentation, which produces NAD plus?
No, so aerobic respiration is occurring only when there's oxygen present. So when there is oxygen present, that then, and the key here is that the reason why, the way we term it is that oxygen is the final electron receptor. And what we mean by that is when that NADH is finally produced, oxygen becomes the receptor for that hydrogen producing H2O. And that's where you have that NAD positive kind of resulting from. So aerobic respiration is different. We would not call aerobic respiration fermentation because the metabolic pathways are completely different. So if we have this kind of, and I do want to be careful, we're using these terms in the very strict, like, biological, microbiological sense. So kind of try to put aside like the terminology as it's used, on roasted coffee bag or, roasted coffee company's website. But the point of fermentation and where the distinction there is that when we have oxygen present, we're going to have aerobic respiration, the TCA cycle, production of 34 ATP, way more efficient. When we have no oxygen available, we're going within that cell structure, within the cellular organism, we're going to have fermentation. And that's going to result in the production of NAD positive that then goes back and is used in glycolysis to produce energy. Hopefully that's clear. I know it's It's
Thank
a
you.
complex, complicated topic. Hopefully I'm explaining it well. So the benefit of anaerobic respiration, let's not use the term processing because I don't want to like enter into this space of like what is anaerobic processing. But so the benefit of anaerobic respiration is easier to manipulate the process, and aerobic would have more complication in processing. Not necessarily, Tristan. The so we just have to be careful that we're not so oftentimes when that term anaerobic is used in processing, you're talking about an environment that doesn't have oxygen. Okay, now there is still some oxygen in water, dissolved oxygen, that does become available to a certain degree for bio, for metabolic pathways. But when we talk about, yes, the byproducts of aerobic respiration being carbon dioxide and water and 34 ATP, yes, are greater than some of these other fermentation byproducts. However, the challenge is that when you have oxygen present, we have, oh, you butchered that question. It's okay. You do have the opportunity for more of your aerobic. So it's your obligate aerobes or other types of microorganisms that thrive in an oxygen-rich environment, acetic acid bacteria, molds, and also your yeast that you have present that are facultative anaerobes are going to be utilizing aerobic respiration to create their energy, not fermentation, because it's way more efficient to utilize aerobic respiration than fermentation. There's a question here. Anaerobic respiration, is it the same as anaerobic processing? I apologize. No, it's not a silly question. Not at all. It's different. And that's where I want to be careful that we're not confusing the two, because when we're talking about anaerobic cellular respiration, we're talking about there being no oxygen available for the cells to produce energy. Oftentimes anaerobic processing is speaking to a oxygen starved or oxygen depleted environment, speaking to and really making reference to the type of microorganisms that we might find in that type of environment. So that's kind of some of the distinction. Okay, cool. All right, any other questions? I know we're like just on time. I knew it was going to go. I was so ambitious. I thought we'd get through this. Yeah, so Anna, she posted this thing from Khan Academy. It's a really good reference. Also, if you want to just watch like go to YouTube, type Amoeba Sisters fermentation. Really cool explanation of it as well. And you know, I think, in fact, if I could like play that and we'd all just sit there and listen to that during class, I would. But it's a great description and understanding of the fermentation process. So, all right. Look for my email this afternoon or here in a few hours with tomorrow's link. And tomorrow we're going to continue to dive into metabolic pathways and then talk a little bit more about specifically fermentation in coffee. Hope you have a great evening, afternoon, morning, day, wherever you are in the world, and we'll talk soon. Thank you, team. Hey, no problem. Everybody have a great day.
Thank you. Thank you.